Everyone. Welcome you all to the workshop. Thanks for joining us to learn about CellProfiler. I'm Suganya Sivagurunadan, a postdoctoral fellow in Beth Simoni's lab at Broad Institute of MIT and Harvard. Today with me, we'll be joining an amazing set of people, Esteban, Paula, and Satavisha from Broad, who are all postdoctoral fellows with me, and Naomi and Cynthia from the ABIC team. They'll all be helping us understand more about CellProfiler. And I thank my Rahmoon from HHMI who helped us organize this workshop. Before I begin the talk, I would like to make sure all of us have downloaded and installed CellProfiler and also have downloaded the materials for the beginner segmentation tutorial. We have also linked another tutorial for advanced segmentation, which the attendees can do at their own pace. Let me give you an outline of how the workshop is organized. We'll begin with discussing why we need bioimage analysis software, and then we'll go over the basics of CellProfiler, its interface, its settings, and the various modules that we can use. Followed by that, outline of the hands-on workshop will be given by Paula Llanos. And then the fun part is the hands-on exercise where we build a pipeline to do a beginner segmentation. With that, uh, let's discuss why we need bioimage analysis software. I'm pretty sure all the attendees in the call would know by now, and that's why you're in the call, but let's get in more detail. When you take a look at the image here, how many of you think it's a colored image? I assume most of you would have thought so. But when you look at it in detail, it is a grayscale image with colored lines in it. Our human brains fill the grayscale parts of the image with colors and makes us believe that it's a color image. Isn't this interesting? Let's take a look at the other example. When you look at the intensities of nuclei in cell A and B, it seems to differ, and so does the sizes of the circles C and D. But is it exactly the case? Actually, the intensity of the nuclei in cell A and B is just the same, and so does the sizes of the circle C and D. This is exactly why we shouldn't believe our human eyes to come up with biological conclusions using our microscopic images. Moreover, we are no longer in an era where we draw down our experimental observations. Rather, we take the advantage of digital cameras to generate images for us. With the help of digital cameras, whatever images that we generate are nothing but just an array of numbers. This makes it more easier for us to depend on computers and softwares to analyze these numbers for us. And being biologists, we'll have a lot of such array of numbers since we take a lot of images. And it'll be more nicer if we automate the whole image analysis process. Let's see why. When we look at the couple of images here, which is from a translocation assay, where the image on the right, you can see the GFP is localized in the cytoplasm, and on the image on the right, the GFP is translocated to the nucleus. By quantification or by looking at it, we know that there's more localization in the of GFP in the nucleus in the image 2 that is on the right. This is the information that you get out of microscopy, which is not even readable with quantifying the mRNA or protein levels with these cells. So microscopic images does contain a lot of information, which complements even other high-end assays that we are trying to do. When we have a lot of images, the whole process can be automated, which makes the process faster and less tedious. And we can analyze multiple images at the same time. We can try to answer a lot of biological questions that we have. And by automating the whole process, we'll be able to distinguish subtle changes that are even missed by our eye. And the main important thing is the whole process will be reproducible when you automate it, since it is all like recorded and down there with the software. By now, we know why we exactly need to use bioimage analysis software. But are there enough out in the field to help the biologists to analyze their images? Luckily, yes, there is. There are amazing team of scientists across the globe who are developing softwares to help make the biologist's life easier. These are some of the open source bioimage analysis softwares that are available. I'm pretty sure most of you would have used Imagery or some of the software that I've listed on the screen. Now let's begin looking more in detail about CellProfiler. What is CellProfiler? CellProfiler is a bioimage analysis software that helps turn your images to numbers. This was developed by a great team of scientists. And it is a free and open source software which can be downloaded and used across different operating systems, Windows, Mac, and Linux. The software has been ranked the most flexible and usable in independent analysis, and no wonder that it has been cited a lot of times in a year. The core idea of CellProfiler is building a pipeline. What do I mean by a pipeline here? We start adding a series of image analysis steps that needs to be applied across images. And the good thing is about CellProfiler is the whole series of steps that you build is applied exactly the same way across all your images. Hence, it is highly reproducible and there's no bias in analyzing your images. We'll begin looking into more about the interface and how the different settings work in CellProfiler. When you download and open the software, this is the welcome screen that you would see. And each of the icons here, I'll go into the details at the end of this talk. We'll go in more details about the settings now. When you open the software, this is how it looks. And on the left, you have the pipeline panel. Each of the lines indicate the different modules. That is an image analysis step. And CellProfiler runs these modules from top to bottom in a linear fashion. And on the right, you have the settings panel. This is where the settings of the corresponding modules appear. If you choose one of the modules on the pipeline panel, the corresponding settings appear on the right. And there is notes panel. If you change a particular setting and would want to make a note about it, you can make a note of it. And if you pass on your pipeline to a friend, they'll understand why you have made this setting exactly based on your notes. And above all, you have these tiny question marks all over the software, which is very helpful. If you don't know what a particular module does or a particular setting in a module means, you can always feel free to click at this question mark and you'll get the whole documentation on what a module does and the setting does in a module. With that, we'll start building a pipeline. The first step in building a pipeline is loading your images. And we have four modules, which we call it as input modules, which will help in loading your images. The images, metadata, names and types, and groups are all these four modules, which will help us in this process. Before that, I would like to introduce you all to a term called image set. Image set is defined as one or more channels at one imaging location or field of view. Again, it depends upon the microscopic image that you have, but you know more about your images, like how many channels does it have, and is it like a 2D or 3D images? But we want to instruct CellProfiler what the images mean, and that is why we need these input modules to exactly instruct CellProfiler to do it. We'll begin doing it, and we'll go through the step one by one. Images is the first input module that you could see. When you choose, the corresponding settings appear on the right. The empty space here, when you right click it, you'll be able to search for your images, or you can drag and drop your images into this panel. And you also have the option to filter your images based on the file type. If you want to filter only the PNG formats or the TIFF formats, that can be done here. And you can also filter the images based on the file names. Once you have loaded in your images, that the image name starts appearing here, like what you see. And when you double click it, a CellProfiler display window pops up. This is how you exactly see the image. And it is very handy that it has a lot of tools. As you can see on the top, it has a toolbar menu. And the magnifying glass that you see here can be used to zoom in your image. And if you would like to go back to your unzoomed version, you can click the back button here, or you can use the home button. And when you hover over your image, the X and Y coordinates of your image appear on the bottom. And you also see the intensity values here, intensity value of the pixels. And there's a scale bar icon that is on the toolbar menu, which you can use to measure length of any features that you see in your image. And the feature appears on the right. And you can also right click the image to adjust the contrast of your image and also to make a histogram appear. Now that you've loaded, we'll go to the next module, that is metadata. Metadata is information that is associated with your images. This is an optional module. If you don't want to use it, you can leave it off. But if you want to instruct CellProfiler about the images that you have, then you can leave it on. And there are different ways you can extract metadata from your images. you can extract it from file or folder names, or you can extract it from, you can extract it from a file where you've saved your metadata, or you can also extract it from file headers. Once you have chosen which method you want to extract the metadata from, you can choose the particular method and you can hit the update button, and all the images and the corresponding metadata gets populated here. When you extract your metadata from file or folder names, there is something called regular expression, which is helpful to extract a certain pattern from your image names. And now moving on to the next module, that is names and types. It is exactly what it sounds. You know more about your images, but you want to instruct CellProfiler about it. That's what we do in this module. You choose a particular image and you instruct CellProfiler to what to call it as. You can assign a name to your images based on image names or metadata like this. If you want to call all of your channel number 1 images as nuclei, or you can do that here. And you can also choose whether instruct CellProfiler about your images, like whether it is a 2D or 3D image. And you assign a name. This is like a sample name that we have given, but you can feel free to assign different names based on your channels that you have. And you instruct CellProfiler about whether it's a grayscale image or an RGB image. Once all these details are given in this module, you click the update button and it gets populated in the place here. And the next module is the groups. This is an optional module. You can leave it off if you don't want to analyze your images into 2 different groups, but if you would like, you always have the option of doing it. So you can leave it on, and you can group your images based on metadata that you've extracted in the previous modules. You can analyze your images based on the well or channel number that you can set it here in this particular module. So with the help of all these 4 input modules, we'll be able to load your images and assign names to your images. If you want to learn more about the input modules, feel free to check out the blog post and a video tutorial that I've linked here. We'll be making the slides available at the end of the talk. And now, once you open the CellProfiler, these 4 input modules exist there. But to do more image analysis steps, you need to start adding modules. Module is the image analysis step that you would like to do. How do you add modules? When you open CellProfiler and press the plus button here, add modules display window appears, and you have the different modules categorized based on all these ways. You can either double click on one of the modules and it gets added to the pipeline panel here, or you can press the add to pipeline, which makes it easier to add it to the pipeline panel. We'll go in detail of the different module categories that are available. You have file processing module, which helps in processing your image input files and also the output. You also have image processing modules, which helps in pre-processing your images. If you would like to crop your image or enhance certain features in your image, those modules are available under this category. You have an option of choosing object processing modules once you've found the regions of interest in your images, and if you want to erode that area or dilate or make some changes to the objects, those modules exist in object processing modules. Once you've identified all of your objects in the images, then you have this measurement modules, which has a lot of different measurements that can be done on the objects. For example, co-localization or the area or size of the objects. Those can be found in the measurement module category. When you've made the measurements, you have a lot of data to display, for which the data tools category would be helpful if you want to display the numbers in charts or histogram. Yes, you have it in data tools module category. And if you're analyzing 3D images, and you always have this advanced module category where you have the modules that work with 3D images. And if you're a person working with C elegance, we have a particular toolbox for that too. If you don't know, like this is a lot of categories, but if you don't know where to begin with, we have a search button on top, where, for example, if you're interested in measuring the area, if you type in area, the module that will be useful to measure area would pop up, and you can add it to the pipeline. And now, more into how it works. Once you've added the pipeline, like once you've added the module to the pipeline, CellProfiler operates in two modes. One is the test mode and the other is the analysis mode. You can imagine test mode as more like a trial and error method. In the test mode, you tweak a lot of different settings. Uh, yeah, you tweak a lot of settings to know whether it works for images. And if not, you can keep redoing the settings. But once you've finalized your settings, you can go to the analysis mode. That's where you have finalized your settings and you run the whole pipeline and you get the data out, like the measurement out. Once you keep adding more modules in there, you see a lot of icons. Um, like each of the lines that you see here is a different module. Module again, meaning different image analysis step that you would see. When a module is underlined, it means the cell profiler is currently in that module. And when you run it using a small triangle that you see here, that runs the particular module. You can run it either that way, or you can also run it by clicking the run button here. When you have a lot of different modules added to your pipeline and when you run it, the whole pipeline runs, like all the image analysis step runs. But if you'd like to just run one image analysis step, then you can use a step button here, which run just one module at a time. If you would like want to skip some of the modules in your pipeline, then you can uncheck the boxes here, which will skip the cell profiler running the particular module. Or if you want to pass at some stage rather than running the whole pipeline, if you'd like to check just if you're on, just check if some of the modules are working fine, and if you want to pass at certain regions, you can use this pause button. And you see there is a small triangle which gives you a warning. And when you hover over the triangle, it will say what warning does the module has to say to you. And you have these small eye icons on the left to the module buttons. Each time when you run the module, the results are displayed. When you want to display your results, you can have the eyes open. If not, you can uncheck it and your results will not be displayed. And you can also see there is a small cross on the left to the module. This means there's an error with your module. If you don't know what that error means, you can always hover over that red cross and you will see the error message getting displayed, with which you can fix the module and then run the pipeline again. And now we have seen all the settings that it gets displayed when you open the CellProfiler interface. We'll begin building a pipeline. We have already loaded the images. Now we'll begin by identifying the objects. Uh, whatever pipeline that I show here is a generalized pipeline. The processing of your images can either be before the identification of objects or after. And again, it depends upon the biological images that you have. I keep mentioning identifying objects, and I didn't even give a proper introduction of what an object identification is. Let's see what it is. We know microscopy images contain a lot of information. For example, in this image here, we care only about the blobs that are present and not about the background pixels. We know what we care about, but we would like to instruct CellProfiler to make it understand what we really care about. The process of instructing CellProfiler on what is background and what is foreground pixel is done by something called a thresholding process. And the result of this thresholding process is what we get as objects. So your actual raw images are your images, and whatever you instruct CellProfiler on what is background and foreground is what we call it as objects. Once you've identified the objects, then we have the option of whether to split these two objects or merge them, and that is the step 2. Now we'll see how the step 1 is done, like how thresholding is done and what is thresholding. Thresholding is a process where you partition your image into foreground and background pixels. And how it is done? Generally, when you display your image in a histogram fashion, you see these high frequency background pixels and also the low frequency foreground pixels. You care about only the foreground pixels more. How do you divide it? If you choose a threshold value somewhere more closer to your background pixels, you could see the identification of objects is done in such a way, along with your objects of interest, some background pixels are picked up as well. This is not how you want. So if you kind of move your threshold values more towards your foreground pixels, now you see your nuclei is picked up, but then you have left out some regions of the nuclei too. You should come up with a value, or you should choose a particular algorithm which rightly picks up your objects of interest. We have different methods to do this, and the method that we have is minimum cross. One of the method is minimum cross entropy. This works for most of the images, which has good signal to noise ratio. And there is another method called ODSU. This method works when you have more than two peaks in your image histogram. And if you want to classify the middle peak as either foreground or background, that's where you use ODSU method. There's another method called robust background, which works well for images which has a lot of non-specific background, or if your images are more speckly. This can be used in cases like fish images. Again, I would like to make a note here. You can never get the perfect segmentation or identification right in all your images because that is how it works. And I highly recommend you to look at the blog post which I've linked here, like when to actually stop trying the different thresholding algorithms for your images. And now we'll see how we can do all these changes in CellProfiler. We have added the Identify Primary Objects module in your pipeline panel. I'll be spending a lot of time in this primary object identification module because this is the initial step and this is the most important module in CellProfiler, the output of which will be used in the downstream modules. So you want to get the first step right. Once you open once you add the identify primary objects, you have the basic settings displayed this way, and you need to instruct CellProfiler about what image it needs to take in and what you want to call the objects as. That's what you give here. The input image name, which you have done using input modules, you can choose a particular image name, and you can also give a name to the objects that will be identified in this module. You can play around with the basic settings like the diameter and of the objects. If you think it did not pick up your right objects, you can always go in for advanced settings. And when you choose advanced settings, there's a whole list of settings that you can play around with to make your object identification more appropriate to whatever you're expecting. The first setting that I would like to go over is diameter range. You can choose the diameter or set the diameter range based on the features of interest. And if you want to consider objects that are outside your diameter range, and if you also want to consider the objects that are touching the edges of the image. And CellProfiler makes it easy for you to understand by giving these colored outlines. Once you identify the objects, you could see there are three different panels that is displayed here. The one on the top is your raw image, and the one that is titled Nuclear Outlines is the objects identified by CellProfiler, and the panel that shows nuclei on the right, those are the objects that will be passed over to the downstream module. When you look at the nuclear outlines image, you see that identified objects has different outline. And what does it mean? The objects that has the green outlines will be passed over to the downstream module. The objects that has the pink outline are identified by CellProfiler, but then it does not fall in the diameter range that you've specified earlier, so that will be missed, passing on to the downstream modules. If you would like to include, you can go back to your diameter settings and adjust accordingly to include this particular objects. And you see also objects that are outlined in orange, which means these objects will not be considered because it is touching the bodies of the image. Again, if you would like to include it, you can change the settings back with whatever we saw in the earlier slide. Once we have, and we have more settings to make the identification of objects easier. We have this threshold strategy, which we discussed earlier. We have three different methods, and you can choose the method that works well for your images. And we also have this threshold correction factor. What is threshold correction factor? If you choose a particular thresholding algorithm and like the different available algorithms and none of it works well for images, you always have this option of setting a threshold correction factor. It helps to nudge the threshold value by making it slightly stricter or more lenient. And I would recommend it always works better when it is around 1. And you also have this option of setting an upper and lower bounds for your threshold when you have like odd histograms, when you don't care about these foreground pixels, but what you care about is somewhere in between, which the thresholding algorithm misses to pick. Then you can set a minimum and maximum value within which will be considered as the foreground pixels. And again, all these settings are helping to make you identify the objects of interest in your images in a better way. There are more settings to help you with that. We have something called threshold smoothing scale and log transform before thresholding. In which cases these work better? For example, when you have image histograms where you can't actually differentiate between the foreground and background pixels, when you log transform this image, then you get a better histogram, which is like where this makes the process of delineating your foreground and background pixels better. We can take a look at an example here. For the images on the right, you could see there's like a varied intensity of nuclei, and you see the nuclei objects are not picked up the way you want it to. When you log transform these images, uh, of course, the image histogram gets better, and you will be able to choose a thresholding algorithm which fits well to choose your objects like in an appropriate way. And now we have identified the objects. We have instructed CellProfiler on what is foreground and what is background. By experience, you know there are like around 8 cells here, but how do you instruct CellProfiler? It is just not one object, but there are 8 different objects. You can take the intensity of fluorescence uh, for your advantage, where the intensity of fluorescence is higher in the middle of the cells and it kind of dips down at the edges. And that is where you can exactly delineate or separate the objects based upon the intensity difference. This can be set in CellProfiler by giving declumping objects based on intensity, or it can also be done using shape. When you're trying to separate objects that are roughly circular, then you can give shape as an option to separate your objects of interest. How this can be done in CellProfiler? You have the option or method to distinguish clumped objects either by shape or intensity. The drop down here will have the shape or intensity, and you can see which works for your images better. And with that, I know that's a lot of information in a short span of time. I would like to know if you have all questions, and we'll be happy to help with that. Else, I'll move on to the next module. I'll give a couple of minutes to see if people here have questions on the primary object identification. I guess there are no questions on primary object identification. I'll move on to the next module that we'll be discussing. That is secondary object identification. Oh, I see a hand raised. If you want to unmute yourself and ask the question.
All right. Thank you. Can you hear me?
Yeah.
All right. Thank you very much for the wonderful presentation. I believe that the hands-on segment is coming towards the end of the whole exercise.
Yeah.
So many people actually found it very difficult to be able to go along because of maybe the speed of giving the talk for now. So I want to suggest that during the hands-on, it should be very, very slow so that people can be able to flow together. Majority of us may be very new to this particular software. So I want to just suggest that during the hands-on, it should be very, very slow so that we can go at the same pace with the beginners. Thank you very much.
Yeah, I'm sorry if I'd been like really fast in my showing over the settings. For sure, the hands-on is designed specifically in a way where you can go at your own pace, and we
All
have
right.
like a PDF linked, and you can go at like each setting one by one at your own pace. I'm sorry if
All
I've
right.
been super
Thank you
fast
so
here.
thank you so much for that.
Yeah. Yeah, let me move on to the next module and make sure I go at a very slow pace now. Uh, the next module that we'll be adding is the secondary object identification. What happens here is whatever objects that you've identified using identify primary objects will be used as seed objects. And in this case, we have identified nuclei in the first step, and the secondary objects is we are trying to identify the cells. Your nuclei will be used as seed objects here, and the outlines, whatever you see on their image here, uh, the colored outlines are very different from what you seen in identified primary objects. The pink outlines that you see are the outlines of the secondary objects, and the green outlines that you see are the seed primary objects. We'll see how to make CellProfiler identify these secondary objects. There are 4 different methods that you can identify secondary objects. Again, the example that we have here is nuclei and cells, but again, it varies. Anything can be primary or secondary objects on, like, it depends on what images you have. The 4 different methods that we have in CellProfiler to identify secondary objects. One is distance n, where your input seed object is taken and a circle is drawn around your input seed objects to a certain distance that you specify in the settings, and it does not depend upon the staining intensity of your images. And the distance b is a setting where a circle is drawn around your secondary prime input objects to a distance where there is staining intensity in your images. And there is a third setting called watershed. Here, you'll be able to come up with the outlines of your secondary objects based on the difference in the intensity, like similar to what we saw in the identified primary objects. And there's another settings called propagation, which is very similar to watershed, but it assumes that all of your secondary objects are of same size. You can choose either of these methods, whichever works well for your images. Once you have identified the secondary objects, we also have a module to identify tertiary objects. In this example here, we have identified nuclei, cytoplasm, and you can identify, nuclei and cells, you can identify cytoplasm as your tertiary object. Again, it depends. If you're interested in looking at the features of the cytoplasm, yes, this is the place where you add these modules. And now, having talked a lot about objects and images, you've identified your objects. You want to make sure whatever objects that you've identified exactly correlates with your raw images. That's where you use modules called overlay outlines or overlay objects, where you can use your raw images and overlay the objects that you identified and make sure that it exactly correlates with the raw images. You can either add these modules to your pipeline or use something called CellProfiler Workspace, which does this iteratively. And now we have loaded your images, identified the objects, and the next is processing. Again, based upon the images that you have, the processing can either be done before the identification of objects to make the identification process easier, or after identification of the objects. Again, everything depends upon the biological questions that you have with your images. What do I mean by processing? Like, you, you can enhance your images or crop them or make a lot of changes to your images. But I would like to make a note that whatever changes that you make to your images, uh, you uh, you should make sure the measurements that you make are on the raw images and not on your processed images. That is one thing to have it in mind. And these are different image processing modules that are available in CellProfiler. Like, feel free to check out the advanced category as well, because we have some of the image processing modules in advanced too. And now the fun part. We have built the pipeline, identified the objects and measurements, the measurement module. The main advantage of CellProfiler is you get a lot of measurements out. And for example, when you add a module to measure area, it is just not area that you get out of this module. You get the perimeter of the objects, the eccentricity, like where 1 represents a circle or 0 represents a line. And you also get information on major axis length or minor axis length, which is the short and long diameter. You also get information on orientation, if all of your objects are oriented in a particular way, and also about solidity. By adding just one module, like, you get all these information. And similarly, when you add a module on measure object intensity, it is just not the object intensity that you get. You also get information on mean, median, and standard deviation of the intensities. You also get the minimum and maximum value of the pixel intensity. We also have a sum of pixel intensities, which we call it as integrated intensity. And CellProfiler also provides information on the upper and lower quartile of the intensity values. For example, if you have identified your objects in the nuclear channel, you can use the same objects to measure the details of that object in another channel. For example, if you've identified, like for example, in a translocation assay, if you're interested to know how much of GFP has translocated to the nucleus, you can identify nuclear objects in your nuclear channel and use those objects in GFP channel to get information on the GFP localization. And we also have modules of measure object intensity distribution. This will be helpful in cases like if you're interested in understanding about to what extent the mitochondria has spread, whether it has spread to the periphery of the cells, then you can choose this module where your cells will be divided into different bins, and the number of bins will be again set by you in the software. And you can, CellProfiler will be able to give information on the intensity values in each of these bins. And you can again choose whether you want to get the total intensity or mean intensity based upon the different settings that are available there, and you can choose. We also have modules for measure co-localization. For example, if you want to see if the GFP translocation has taken place, of course, you see more of co-localization in the images from the images where you see the GFP translocation. And now you've built the pipeline, you've made all the measurements. How do you export the data? We have modules called as export to spreadsheet and export to database. The export to spreadsheet option saves your measurements in CSV format, and export to database is useful when you have want to analyze it in another software such as CellProfiler Analyst. And I would like to make a note that when you run a CellProfiler pipeline, CellProfiler does not save any of the images. Like if you would like to save the overlaid objects on your images or any other changes that you make, you would like to save them, always add a module called Save Images that will help you save the images. And you can also save the whole projects as CP project files, which saves the whole pipeline along with your images. But if you would just like to save the image analysis steps, please feel free to export the pipelines as CP pipe. This does not save your images, but saves the image analysis steps that you have. And I would also like to point out that CellProfiler can be run with other open source image analysis software such as CellPose, ImageScript, ImageScript, or Stardust. If you want to combine both together in a single pipeline, we have plugins available for that. I've linked the plugin details here. Feel free to check out if you're using any other softwares along with CellProfiler. And I know that was a lot of information in a short span of time. If you don't know where to begin with, we have this welcome screen, and each of these icons would make you help understand more about CellProfiler. When you click the try example, CellProfiler loads an example pipeline, and you can go through the modules one by one to understand what it does. And we also have this getting started. After loading the pipeline, you have different steps instructed here to know where to go and what to look for. And if you want to know how these pipelines can be applied for your experiments, we have different published pipelines that are available on the web page to see what are the implementations or where this can be or which assays can be done using the CellProfiler pipelines. And we also have links to a lot of tutorials, like we have just provided links to beginner segmentation and advanced segmentation, but there are tutorials available for a lot other ways CellProfiler can be used. And if you're building a pipeline, if you're stuck at some point, or if it's nothing about CellProfiler and you have a general image analysis question or a general imaging questions, we have something called forum.image.se, where you can post your questions, and all the community partners from all the softwares that has been available for bioimage analysis softwares will be there to answer your questions. How do you post your questions? When you go to the Image SE forum, you have an option of choosing a new topic, which allows you to post a question. Once you choose this, you have a template displayed where you can fill in the details of the images that you have and what you've done so far with your images and what you're trying to understand, like some background of your images and what are your goals that can be input. And the questions will be answered by all the community partners. With that, I would like to thank Center for Open Bioimage Analysis, which has made all these workshops possible, and also the Simony Lab members, Carpenter Singh Lab members, and Leong from HHMI for this opportunity to present, and also Maya Rahmoon, who has made this was very helpful in organizing this workshop, and Naomi and Cynthia from ABIC team who helped organize this workshop and will also be teeing today for the hands-on exercise. With this, I would like to know if there are any questions, or I'll hand it over to Paula, who will be going over the outline of the hands-on exercise that we'll be doing today.
Okay, super. Thank you, Sugania. Good morning or afternoon, everyone. I am Paula Llanos. I'm a postdoc also in the Simini lab. I will share my screen now. Here I see. Okay. You can see my screen. It's okay. Yes. Okay. Super. So I want to go through the exercise that you are doing today. So the main aims for this exercise and tutorial that you will follow, the first aim is segment some parameters for larger parent objects, will be nucleus, cells, and cytoplasms. So the first aim will be segment those objects. The second aim will be segment some organelles within the cell compartment that we already segment in the first part. Will be that organelle that we are segmenting is the nucleoli, which will be inside of the nucleus. And in the end of the tutorial, you will be able to get some morphological measurement of the organelles. So what kind of image you will use during this exercise? We are using this, sorry, we are using the U2OS cells, which is a cell line for osteosarcoma. It is of those cells are imaging in 5 channels. This is the data bank where we obtain those cells. That image is contained in the image folder. Everything is described in the tutorial. So the same that I'm saying here, you will also find in the tutorial written. So in that folder with the image, you will find 50 images, which is 10 site images in 5 channels and represent 5 mock treat wells from a single 384 well plate experiment. And so the first step will be drag the image in CellProfiler. And when you drag the image in CellProfiler, you will find here some files, which are not TIFF files, which is the more common format for image. You will find this MPY image, which are illumination correction that was calculated previously. The illumination correction is something that you can use, and it's more to avoid the shading artifact producing for the microscope. You can apply, I mean, it is in this case will be necessary to apply that illumination correction, but it is optional, depends what kind of image do you have in the moment that you are using. So but those files was already calculated previously. Okay, so we not just need to apply the illumination correction, which is those files. If you open that file, you will see an image like that, which is that represent the shadow that the microscope produce. So that is the shadow that we wanna try to correct in our set of image to have more an even intensity image. Okay, so not worry very much about that part because this already was calculated. And when you open the start pipeline, you will find this module already loaded, which is correct illumination apply. So it is what is doing here. After that, when you finish your the exercise, you will and if you follow the step right, you will include those modules. So I just categorize the modules, then you can have an idea about what you are doing. So this, it kind of, I categorize the modules in 3 categories. The first category will be like to segment the nuclei, cell, and cytoplasm. The second category will be segment and get the organelles, the nucleoli in this case. And the third part will be the modules that allow us to get all the measurements that we want to obtain from the object that we already segment in the previous steps. And in the end, we can also export all the measurement in one single CSV file. Then you can check in your computer after to analyze your data. So in this first, in the exercise, as I say in the tutorial, everything is very well described. So don't worry about uh, to take very much detail here. I just want to explain you how is the pipeline, how works everything, but you will have everything in the tutorial. Okay. So in the first module is identify primary objects. So this is this as Suganya explained very well. So we will just segment the nuclei, and you need to adjust some parameters here to get the proper segmentation of your nuclei, which is important because then will be define each cell in the end, right? This primary object is kind of the seed for what that will relate each nuclei with each cytoplasm and with each cell. And then we will include a secondary object, the identify secondary object module, which will allow us to segment the cell object like that. So this is in this case, you will find an image where you can see the nuclei that was segmented in the first module, the identify primary object, and also the one that you segment here. And if you are happy with the results, you can continue, with the next module, which is identify tertiary object, which is just basically to get the cells that we already segmented and take out the nuclei, and then you will have as a result the cytoplasm. So then we will have the 3 main compartments of the cells. In this second part of the module that you are working on, you need to work on a little bit of the pre-processing for to can get the nucleoli inside of the nucleo. Okay, so this module, the enhancer surprise features, mask image, are for to help you to can segment better your nucleoli inside of the nucleus. So once you have this pre-processing step, you can have also the identify primary object to segment the nucleoli. And here in this module, which is the overlay outline, you can overlay your raw image with the object that you already segmented. So for example, here is my raw image and also it is show the nucleoli inside of the nucleo. So if I am happy with that, you can also continue with the next part of the pipeline. This is kind of a sanity check. You can include this module many times as you want. You can also include here the cell shape to check if all the objects that you want to segment in the image are okay or not. So in the next module, in the next set of modules here, is more measurements modules. So the idea here is to get measurement of the object that we already segment, right? The nucleoli or cells. So what you need to choose here, which is very important because we segment the main compartment and we want to measure something in those compartments. But what we want to measure is the signal, the original signal, because the pre-processing that we did to obtain the object, we modified the intensity, modified the Instagram sometimes, but it was just to obtain the object itself, right? The area and the compartment. But what we want in the end is measure the original signal, right? We don't want to measure a signal that is not coming from the microscope itself. So here, when you select the image to measure, you need to select your original image and not the image that we produce to get the segmentation of your compartment. And finally, here you can select the objects where you want to measure those signal. You can also include nucleoli, for example, if you want to measure also the object, the intensity inside of the nucleoli. So it's more up to you. So, you can play a little bit with that. How looks the measurement, the measure modules? It looks like that. That will give you an average of all the objects that you measure in that image. So where you will when you export those measurement, everything, you will find more detailed info, the data, but you will get when you export the spreadsheet this in the end. But if you just want to check, for example, here some statistical about what is measuring in the image that you're analyzing, you will find something like that, which is the more average. And so we know that what happens, for example, if I don't have fluorescence image, I have more histological imaging with other kind of dye, right? So here in CellProfiler, you also can analyze histological imaging, tissues, and everything. So here I recommend in the website of CellProfiler, you can find tutorials with some examples. So I recommend also to use this tutorial in the website, which show you more details about how to work with this kind of image. And in kind of briefly, it is what the module that is very useful. The module that is very useful in this case is a mixed color. So with these modules, you can kind of separate the kind of dye that I'm using in my stain in different channels. So the treatment is similar to what we did in the fluorescence, right? We separate in different channels, but with this module, is able to recognize the different dye that we are using to can separate, and then you can continue segmenting in the way that we already was doing before, right? The module that we are already using. So for tissue and everything, I recommend to use this module first, and then continue with the rest of the module that Sugani already explained. And also here in this module, you can find, as you see, different staining that is common used in this kind of tissue, so and in tissue in general, right? So you can even just select the kind of staining that you are using and then just separate the different staining in different image. So now will be your turn. So what is happening now? You will be assigned in a breakout room. So it is very encouraged that a person in each breakout room share the screen. Then everyone is working together, is following the step in the tutorial. Follow the step described in the tutorial. Each step suggests a time to spend on it. Okay, so it has approximately time. And all teaching assistants will be going to the breakout rooms to answer questions. So you can also raise hand if you have something, you need more help. But in general, all the room will have a teaching assistant to help you. Also, after you assign to breakout rooms, we will have a 10 minutes for break, so you can go to get a tea, and then, or if someone, it's okay, you can just read the tutorial. Meanwhile, the rest are already prepared to start the exercise. So any questions so far?